So it's my great pleasure to introduce our next speaker, who is Professor John O'Keefe. John did his BA in New York and then a PhD at McGill, and forever has been part of UCL. And when I arrived 4.5 years or so ago, I knew John in his role as the Foundation Director of the Sainsbury Wellcome Centre, which was a secured enough money for a new centre, a new building in neuroscience, a beautiful new building which has recently been opened. John has a hyper-distinguished career in neuroscience that culminated in 2014 with the award of the Cavell Prize in Neuroscience, and then in the Nobel Prize for Physiology or Medicine for his work on plate cells. And I'm delighted he's here to present to the association. So, John, thank you.
Thank you very much, David, and let me say I'm delighted to be here. Thank you very much for inviting me to talk. I'm going to tell you about two things. I'm a basic researcher, and I've spent most of my career at UCL trying to understand the basic mechanisms of operation of a part of the brain situated in the temporal lobes here. But like I think many, perhaps most basic researchers, I've always at the back of my mind hoped that someday we'll be able to use this information to make a contribution to human health. And so in the second part of my talk, I'll tell you about our beginning research efforts in that direction, and particularly how we're trying to use our understanding of the mechanisms of action of this area in the temporal lobes to understand and address the problem of a major problem of health, which is Alzheimer's disease. So let me begin then at the beginning. Let's see if I can get all these things to work. Which is, it's going to be a little difficult here. Which is the story of how we began to think of this part of the brain as having a major role in memory, and particularly a particular kind of memory, episodic memory. Early in his life, the patient HM, or now since his death, we now call him Henry Mollison, suffered from epileptic fits, and as a result of that, was subjected to an experiment, frankly experimental operation in the early 1950s, which involved the removal of two large bits of brain on both sides of his brain, shown here from the ventral view and here from the frontal section. And essentially, what the surgeon did was go in and take out large parts of the mesial part of the temporal lobe. And you can see this is the intact brain, and you can see here. And essentially, this area is rather complicated. There are many different structures in that part of the brain, but the major structure is this area, the hippocampus here. And Brenda Milner, over many, many years, together with her student, Sue Corkin, studied HM very intensively to see if they could identify exactly what the problem was. The operation, to some extent, ameliorated the seizures that HM had. But when he came around from the operation, it was very obvious right from the beginning that he had a profound deficit in his capacity for memory. He essentially could no longer store what we now call episodic memories. He could no longer remember what he had for lunch, who he had talked to the day before, where he had been. He could no longer store the kinds of memories which represent the everyday occasions of your life, your day-to-day life, what you did, who you did it with, where you did it. And they summarize, Sue Cork in this very nice book, summarize the deficits that they felt he had. He cannot recall anything that relied on personal experience, such as a specific Christmas gift his father had given him. He retained only the gist of personally experienced events, the general sense of the things, plain facts, but no recollection of episodic memories. So when I started researching in the brain in the late 60s and early 70s, and particularly when I came to UCL, having, as David said, been at Montreal, where in fact Brenda Miller had been one of my teachers, I saw this as an opportunity to see if we could actually use newly developed physiological techniques in animals to see if we could see what memories looked like. Could we look at the physiology of memory by studying this part of the brain, the hippocampus? So this is the hippocampus in the human, and here it is lying in the mesial part of the temporal lobes. And that's one of the areas. There are other areas that were removed in HM's, from HM's brain, but people like Brenda Milner thought that they knew what the amygdala did. It had something to do with emotions, and therefore they, by a kind of neural subtraction, decided that the major source of his difficulties came from the removal of the hippocampus. Here's what the hippocampus looks like in a rat, or in a very nicely colored stain. And it really is a very simple anatomical structure. It consists of several parts here called the cornu-monas fields, plus the dentate. And then there are these ancillary parts of the hippocampus, about which we knew very little back in the 1970s, but which turn out now to be very, very important. So what we did was we essentially put electrodes into the brain of animals during an operation, allowed the animal to recover from the operation, and then monitor the activity of these cells, particularly these cells here, to see if we could understand how they participated in simple memory tasks. And over a period of months, my student Jonathan Destrovsky and I tried lots of different memory tasks. And very clearly dawned on us, it became very clear very soon, that we actually weren't making much progress. So we decided to forget about the memory task. We decided to forget about HM, and decided to just go back and do a more ethological study, a more ethological approach to what these cells were doing, hoping in the end that their function and therefore perhaps their relationship to memory would emerge from this study. So we just started looking at when the cells fired as the animal went about its daily tasks, as it went about eating, drinking, and so on and so forth. And what we eventually began to realize is that the cells weren't interested in what the animal was doing. They didn't care about why it was doing it, what its motivational state was. They began to be interested in where the animal was doing the behavior. And so when we started to look at it, we saw that, for example, this cell here was interested when the animal went into this part of the box and not to other parts of the box. And you can see this here. The cell doesn't fire, as shown in this histogram here. It doesn't fire when the animal is in most of the box, but it fires when it goes into this area here, A, and a little bit B. So on the basis of this, we thought, well, maybe this is providing the animal with some sort of spatial information, telling it where it was doing things, where it was when it was doing things in the real world. And what now is considered to be a rather bold leap of imagination, we said in this very, very short paper, these findings suggest that the hippocampus provides the rest of the brain with a spatial reference map. That it's providing something that had been suggested by a Californian psychologist several years earlier, a man called Tolman, which was that when animals learn their way around an environment, when they learn to go from one place to another, they don't do it by following a simple route. They don't say, I go down to the corner, turn left, then go up to the next object and turn right. They learn it by forming a map of the environment so that they can actually address that map, very much like an ordnance survey map, when they want to find their way around. Well, I then got together with a man called Nadell, and on the basis of looking at the literature that was existed on the hippocampus at the time, we concluded that it might actually be forming this map, and we could see it in much of the effects of, for example, damage to the hippocampus that had already been recorded. And I won't go into it in any great detail, but what we thought was that the map might look something like this. It might consist of a set of place representations, these cells we had actually seen, so that every time an animal went to a particular place, there was a set of cells in the hippocampus which actually became active. And so when it went to place A, there was one set. When it went to place B, another set, and C, so on and so on. And in addition to that, if you had a map and not just a set of place representations, you need to have some way in which the cells which represented the different places were connected together. For a map, you need not only know where you are, but how to go from where you are to other places in the environment. And the easiest way to do that is to set up a set of representations which represented the distance and direction from where you are to the location that you want to go for. So we imagine that there would be a bunch of places, and they would be located relative to each other by these distance and direction vectors. And then we came up with a whole bunch of predictions about what would happen if an animal actually lost this mapping function. And so we said animals can use the map to locate themselves in an environment, to locate items, including rewards and punishments in that environment, and to move from one place to another by any available route. One of the nice things about a map is you're not confined to any particular route. If you want to go from A to B, for example, to go from here to here, and you find that that path is blocked, then the system should be able to, as a map can, generate alternative routes, AC to BC. Perhaps even using something like vector algebra. You can do detours, you can do shortcuts. So we made suggestions. A good theory actually makes very clear and very concise suggestions. We suggested that there should be other signals, not only just these place representing signals, but signals which would represent distances and directions. At the time, there was no evidence for those sorts of signals in the hippocampus or elsewhere. And also that if the animal lost that representation, then it should no longer be able to do place learning. It should not know where it is, and it should not know how to go from where it is to other places. And what I'll show you in the next couple of slides is that essentially, over the last 40 years, we and many, many other laboratories have essentially confirmed those general predictions. We've found place cells, the directional and distance cells, and we've also shown that animals with damage to this part of the brain actually have deficits of the sort that we predicted. So the first thing that we needed to show was that animals with damage to this part of the brain were no longer able to use place types of navigation, navigation using what would look like reading a map. And the chap who really did all the work on this is Richard Morris, who's at Edinburgh. And what Richard said was, well, if you're right, and I think he thought at the time I probably wasn't right, and was trying to disprove the theory, which is exactly what you want people to try, then we should be able to devise a very simple behavioral task, which will actually throw up the ability of the animal to use this mapping structure. And if this part of the brain is involved in it, then the animals with damage to that part of the brain, the hippocampus, will not be able to do it. And what he came up with was a very, very simple task. It's called the Morris water maze, which essentially is a swimming pool for rats, or more recently, mice. And what you do is you place into that pool, it's full of milky water, so the animal can't actually see anything underneath the surface of the water. You put into that place a small hidden platform. Then you put the animal down into the platform, into the water maze in different locations, and its job is, after it swims around a bit and says, oh, this is nice, and it goes a little swim, but to find the platform so that it then can get out of the... and it doesn't take them very long. If you give them about 8 or 10 trials, very quickly, normal animals can find their way out. So here's an animal the first time he's put in, he wanders around until he finds the platform, and here's an animal after the 8th or 10th trial. They learn very, very quickly. And it turns out that damage to the hippocampus prevents the animal from learning this. They essentially continue to swim around for long periods of time without finding the platform. So I thought it would be useful to see what this looks like in reality. And here's the water maze. This is an animal which is well trained. Here's the hidden platform, and here's an overhead view of the whole task as the animal, so that we can actually quantify the animal's behavior. And he gets up and he stands on the platform. Now, of course, being psychologists, you want to make things a little more difficult, so you push the platform down so that when he gets there, he started from a different location. When he gets there, he actually doesn't find the platform there, but he shows a very interesting behavior. What he shows is that he gets to the location, here it is here, shown by this aneurysm, and he swims around in the location. So this is called a probe test, and it tells you an awful lot about what the animal is doing. He's not going there because the platform is there, but he knows that that's the location on the basis of the location of the platform in the room relative to the cues in the room. And this turns out to be a very powerful test of hippocampal function and has been used now for about 30 some odd years to test hippocampal function. You can't go into a major laboratory in the world, or a behavioral laboratory, or to any drug company without finding a water maze there. And Richard still wishes he had patented it with water maze. Right, okay, so we also know, through work in our lab and other labs, that there are all the signals that we would have hoped to find to construct the mapping system. We know that there are not only place cells, but there are, in addition, here are the place cells, but in addition, and not necessarily in the hippocampus, but in these areas surrounding, these retrohippocampal areas, which are actually projecting into and receiving information back from the hippocampus, we know that there are cells which represent the animal's heading direction. So they don't tell the animal where it is, they tell it what direction it's heading. And there are also cells which look like, although we're beginning to find problems with this interpretation, I won't go into that, which look like they're providing the metric for the map. You not only need to know the distance between two locations, but you need to not only know the direction, but the distance between two locations. So let me show you a little bit of what these cells look like to give you a flavor for So here's one of these place cells, and we now have lots of technology to help us quantify what these cells are doing. So we do things like put the animal into different shaped environments, such as circles or squares, and we can watch where the animal is. He has little lights on his head. We can watch where he is as he goes around the environment, so we can tell that he's been in lots of different parts of the environment. And then we can also tell where he is when the cells fire. And this is a cartoon we've made, which is, and what you hear are the spikes firing from a particular cell in the hippocampus. And as you can see, the animal goes all over the place. He's looking for little bits of food. He's slightly hungry. He's looking for food that's thrown in at random. But it only is when he goes into this location here, as shown in this heat map, that the cell fires. If you look at other cells, they fire in other areas. Another one will fire over here, another one will fire over here, and so on and so forth. The collection of cells seem to cover the entire environment. So these cells can actually present the information, represent the information, which actually tells the animal where it is in the environment. And people have actually shown that if you take these cells, you can, as it were, predict where the animal is just on the basis of the cell firing. So they're a very good clue as to where the animal is. About 1984, a man called Jim Ronk, working in New York, found the cells that I had actually been looking for. I spent the next 10 years having found the place cells looking for these other kinds of cells. Where are the cells which tell the animal what direction it's facing in, how far it's gone from A to B? And I didn't find them because I stayed in the hippocampus itself. And it turns out that they're located in these other retrohippocampal areas here, behind the hippocampus, and the information is being projected into the hippocampus from these cells. So here's one of these head direction cells. And these cells don't care where the animal is. As the animal goes around, I put little arrows here to represent when the cells fire. And you can see it doesn't matter where the animal is so much. All it really cares about is that the animal is heading in a north or slightly northwest direction. So if you sum up all of these arrows and over time, you find that the cell is essentially telling the animal you are pointing in the north-northwest. One of the interesting things is that a whole bunch of these cells do the same thing as the place cells. They capture the distribution of possible heading directions that the animal can take. And if you manipulate things, spinning the animal around, confuse it or something, so that you rotate one of these cells, all the others rotate together. So the system forms, the cells form a system which actually act like a compass and represent where the animal is heading at any particular time. And then finally, recently, two of my students who went, Norwegian students who have a lab in Trondheim, showed that there's another kind of cell called the grid cells, which form not just single fields, but a whole set of distributed fields, which look like this. Here's the animal running around frenetically. And you can see the cell doesn't fire every place. It fires in these areas here, shown in this heat map here. And if you look at this pattern, what you can see is that the firing of the cells represent the apices of equilateral triangles. It represents this beautiful pattern, which it's very easy to see that you could use this firing pattern to tell you how far the animal has gone in any particular direction. So if the animal is moving in this direction here, then the cell will say, you have now gone 3 bursts. So we think we have all of the ingredients for constructing the cognitive map. And we have done a lot of work, and ourselves and others have done a lot of work to try and understand, for example, what are the inputs to tell a place cell where the animal is, or and so on and so forth. And over the years, we've tried very much to, for example, see whether these cells are responding on the basis of the cues that are available to the animal in the environment, or whether they're using self-motion information. Is the animal saying, I'm now here, and I know that if I walk in a certain direction for a certain distance, then I can activate the next representation. And to get at that, we recently have been using virtual reality environments where we put the animal into an artificial world, and where we can control all of the information that the animal has, and we can also monitor its moving. So here's a mouse in this case, sitting on a floating ball. And as the animal runs, the ball turns, and we can monitor the animal's movement. We also can tell the animal where it is in this artificial environment by projecting things onto these VU screens. And when you do that, what you see is something that looks like this. And this is a screenshot from an animal looking down this corridor. And I'll show you what it looks like if he actually moves in the corridor. And you'll hear the firing of one of these cells, or not. Okay, that's not working. So essentially what the animal does is he runs from one end of the corridor to the other, and then at the end he gets a reward, and then he's transported back to the start of the corridor, and then he runs down again. So we can see where the cells fire relative to these cues. And then we can say, are these cells firing because of these cues here, or are they firing because he knows he's at the start and he can count steps and activate the next set of cells? And it turns out the answer is both. If we take away the visual cues, as shown here, then a cell which fires in the middle of the track, as shown by this a bright red spot here, stops doing so. And it looks as though the animal is using the cues on the side of the track to tell it where it is. But in addition to that, we can also show that it's using its own information from its own movements. So if we put the animal down in let it run and find out where the cell fires, as this one is doing, and then take away the cues, we find that the cells don't fire, as we'd expect. But if we do the following thing, put the animal down at the beginning of the track, and then as it starts to move, take away the cues, so it no longer has the visual information, we find that many of the cells still continue to know where to fire because the animal can count its steps. So the cells seem to be able to say, I know I'm here, and now as I run 4 or 5 steps, the cell should fire. And so we can show that both these types of different information are operative in the whole thing. Now, this is all very well and good, and We think we understand quite a bit about how the system works in rats and mice. What has this got to do with humans? And of course, you'll recognize that there's a problem with studying spatial navigation in humans, because how are you going to, for example, do imaging studies as an animal, as the human is wandering around in a large environment? And the answer is, of course, what you do is you use virtual reality again. And in fact, we used the virtual reality for studying the human hippocampus before we translated it to the animal world. And the work that I'm going to tell you about has been done mostly by Neil Burgess and Eleanor Maguire. And here, Neil is the head of the Institute for Cognitive Neuroscience here at UCL, and Eleanor is part of the Wellcome Trust Imaging Center. So realizing that we could use virtual reality to study activations of the hippocampus and get people to wander around and navigate in fairly large-scale environments, what Neil did was he took apart one of these shoot-'em-up, first-person shoot-'em-up computer games. This is now back in the 1990s. And essentially, this is a game in which you went around this very complex environment, and every once in a while, a monster would appear, or some other disreputable character would appear, and your job was to shoot it. So Neil, who has a PhD in mathematical physics, spent about several weeks and months taking out all the disreputable characters, all the guns, and all the monsters, and so on and so forth, so that we were left with an environment which was a fairly large environment, 70 by 70 meters, through which people could actually learn about the environment and through which they could navigate. And here are a couple of screenshots from it. There's something for everybody. There's a pool hall here, a bar, a modern cinema. And of course, this is the outside of it, and you can move along these routes, or you can move inside the buildings. And the essential thing is you can go from any one location in this environment to any other by lots of different routes. And you could test how good people's navigation was. So when Eleanor looked at the task in which she trained people to go from one place to another, and then while they were doing that, scanned their brains, she found that there were some people who were very good at going, for example, A to B, and then there were others who could also do it, but weren't by circuitous routes, and then there were some people who got lost. And if you then plotted the activation in the hippocampus, shown here, as a function of how well they were doing the task, she found that there was a pretty good relationship. And this is a PET scan, so it was blood flow versus accuracy. So some people were very good at doing the navigation task and had high rates of blood flow in the hippocampus, and others were not so good and had lower rates. And it turns out that the hippocampus and another part of the brain, the parietal cortex, are the only parts of the brain which were actually activated in that test. Eleanor then went on to show that in this really famous study that if you look at expert navigators, London taxicab drivers, they actually have a bigger part of the brain, the hippocampus, than others. And interestingly enough, that increase in size is a function of how much experience they have in finding their way around, navigating the streets of London. So the longer you've been a taxicab driver, the larger your hippocampus. And when you retire, it goes back. You're not born to be a taxicab driver. You acquire a large hippocampus. Okay, so let me take some time now. How are we doing? Okay. To say what we've been thinking about and doing recently. Because it turns out, and we didn't know this when we first started working on the hippocampus, but it turns out that if you look at the sequence of the development of pathology in Alzheimer's disease, both the development of the plaques and the tangles, and particularly the tangles, as has been done by Brock, what you find is that it looks as though the first expression of the disease is in this area, in the temporal lobe, the hippocampus, and particularly in the entorhinal cortex, the area containing the grid cells. And as the disease progresses, it looks as though it spreads out from this area and to other parts of the cortex until it finally takes over the whole of the cortical mantle. And so the idea is that perhaps it's spreading from a particular locus, a seated area, and it's spreading perhaps along even neural fibers, we don't know for sure, to other parts of the brain. So thinking about this, we thought, okay, can we use our knowledge of the function of these parts of the brain in rats and mice to begin to study what goes wrong in an animal model? And I'll show you some data from that. And can we also begin to see if we could come up with tests, particularly spatial tests, which would enable us to pick out this very, very early stage, the MCI stage, or even perhaps earlier stages of the disease process in humans. And I'll give you a little flavor for where we're going with this. In a study that was done by Francesca Kikuchi and Tom Wills, we used a mouse model, an early mouse model of Alzheimer's. It's not a complete model, of course, and none of these models are really completely replicate the whole disease. This is an overexpressor of APP. And we showed that in a simple T-maze task, a task which we know is sensitive to hippocampal damage in normal mice, these animals actually showed a deficit. And the task is very simple. The animal is put down in this part of the box. It goes down to the end, the T here, and it has to go right because it can't go left, as it does here. And then, after a delay, it's allowed to make a choice, and its job is to go in the opposite way. And there's a natural tendency to do this, but we reinforce that by rewarding the animal to go there. And it turns out that the animal's performance on this is when we looked at these transgenic animals, is related to the plaque burden. So as the animals had more plaques as they got older, their ability to do this task was particularly impaired. And here's some more data showing that, especially if you introduce a delay, then the animals, not when they're young, as shown here, but when they get older, showing that this is a function of age as well as the mutation, they begin to get very much worse at this. Interestingly now, if we look at the place cells in these animals, what we find is that the place cells are normal in the younger animals, but as the animals get older, they begin to show all kinds of deficiencies. So here's a bunch of place cells. As I said, they fire in different locations in wild-type animals, and here's the cells in this transgenic animal, and they look fine when the animal is young. But as the animals get older, again, the wild-type cells are pretty normal, and some of the transgenic animals had cells which were normal, but some of them had very, very poor, deficient cells. And you can say this guy here, he's very bad at localizing where the animal is. It looks as though the ability of these cells to tell the animal where it is are beginning to become deficient as the animal gets older. And in a recently published study, I won't go into this any great detail, the same thing has been shown for the entorhinal grid cells. This is a different animal, a different mouse model. It's an animal expressing mutant tau, and you get this lovely buildup of tau here with time, and you see the same thing in that animal. You see that the animals in the water maze are very deficient when they're older, but not younger, and also in a T-maze task, they also show deficiency. So we think now, the beginnings of the ability to look at behavioral and cellular deficits in these animals during the progression of disease. And of course, what we'd like to see is that at an early stage, the grid cells are impaired, and it's only then at a later stage that the place cells become impaired. Okay, and then, and as I say, there's deficits in the grid cells at a late stage in these animals. Here are the grid cells at a very late stage, and they're much worse in terms of their ability to represent the environment. And then finally, as I say, we would like to begin to develop, on the basis of our understanding of this hippocampal function in animals and in humans, like to develop sensitive tasks for spatial memory and spatial navigation to see if we could begin to pick up the earliest signs of pathology. And it turns out that of course, Alzheimer's patients show deficits in topographical disorientation and finding their way around. Granny begins to start to wander and not find her way around. So Neil Burgess, in collaboration with various people at Queen Square and elsewhere, including Varga Gedem, Lisa Ciablati, and particularly Dennis Chan, who's now at Cambridge, have been working on trying to develop these tests. This is one of the first ones that they developed, and it's actually looks like a very powerful test. It's based on one of Piaget's tests, which is, can you identify the same scene from different orientations? So what you do is you show the person or the patient a set of mountains. It's called a four mountains task, as seen from a particular scene. And then after a very, very short period of time, 2 seconds, you show them 4 other pictures, only one of which represents the same group of mountains, and the other 3 are foils, which are other groups of mountains in different arrays. And this is the correct one. It's seen from a slightly different orientation than that one. When you do this, and you study hippocampal patients, as shown by this group here, and these are patients who don't have problems with visual perception, visual memory, what you find is that they all are very, very bad at doing this. They're very bad at doing the task, and in some cases, they're bad even when all of the pictures are in front of them. You don't even have to make it into a memory task. You can make it into a perceptual task. And it turns out that this task is pretty good at picking up both AD patients and MCI patients relative to controls or other kinds of demented patients. And this is even when you look at MCI patients with and without biomarkers. So we're very, very optimistic that this is going to, these are still small numbers, but we're optimistic that this is going to provide us with a tool for beginning to see the first stages of disease. And there's a nice correlation between deficits on this task and hippocampal volume. One of the standard assays, of course, for the early stages of hippocampal for Alzheimer's is to actually just measure the cross-section of the hippocampal volume. So let me summarize then. I think what I've told you is that we know a lot about the hippocampus in humans and animals. It provides a cognitive map of environments which allow the animals and humans to navigate. There are 2 different ways. I didn't go into this in any great detail, in which you can localize yourself within the map. You can look around and you say, well, here, the doors are up there, and the screen is over here, so that must tell me where I am. Or, once having done that, you can, as it were, use your own inertial internal navigation system, close your eyes, and say, okay, if I rotate in a particular by a particular amount and walk in a particular direction, I will activate other places. I will know that I'm now in this. We're pretty sure that a similar system exists in humans. It's doing slightly more work. It's providing slightly more information. It provides, we think, in an episodic memory, it enables you not only to know where you were in the past, but when you were there and what you were doing there. And we're beginning to use that information to actually begin to see if we can study both the biological processes that go awry in animal models, but also to develop tests for humans. And let me finish by thanking the various funders. The Gatsby and the Wellcome have funded us for a long time, and they've, as David says, they've combined to provide a substantial amount of money to set up the Sainsbury Wellcome Center at UCL, a center for studying the way in which neural networks in the brain actually interact with each other to support cognitive and emotional and other processes. And let me end by acknowledging UCL. I've been here. It's been my home for almost my entire research career. It's a great place to be if you want to do basic research. It's also a great place if you want to translate that basic research into ways of improving human health. Thank you very much.